Hello everyone, today we're going to talk about one of the favorite creationist tropes that concerns genetics but doesn't actually bother to deal with genetic data. So, let's jump right in. Virtually every Christian creationist adheres to the rather cynical doctrine of original sin. According to them, after Adam and Eve ate from the tree of knowledge of good and evil, everything went to heck. Quite literally. This happened because God became really upset when he found out that Adam and Eve disobeyed his orders to not eat from the tree. Considering for a moment that Adam and Eve were created without the ability to know that disobeying God was wrong, forbidden knowledge they could only acquire by eating the fruit, and considering that God himself placed the tree, including a deceiving snake that was more clever than all the wild animals, right alongside Adam and Eve in the Garden of Eden, the events that followed from this setup shouldn't be all that surprising to anyone, even if they aren't omniscient. Anyway, as punishment, God cursed Adam and Eve as well as pretty much everything else. All the animals and every human yet to be born, for some reason. This curse is often called the Fall, the reason for the existence of things like sickness, death, and painful childbirths, for some reason. Creationists are often saddled with the misconception that evolution means everything is getting bigger, better, stronger, and smarter, and they often will contrast this with their belief in the fall, where everything is getting worse over time instead. That's why they are often quick to point to harmful mutations to discredit evolution and bolster their own belief in the fall. And there's this idea that became popular among creationist circles which ties in neatly within this line of thought. I'm talking about genetic entropy. This topic has been covered in detail by other YouTubers such as Paleogenomic and Tony Reed. However, our 2 cents on it are a bit different from theirs, so that's why you're getting it here. Now, genetic entropy is the idea that populations of organisms are undergoing genomic degradation over generations, leading to the decline of fitness, meaning they become less reproductively viable and less able to survive in their environment as time goes on. It asserts that while very harmful mutations are readily eliminated from the gene pool, natural selection is not effective enough to stop the influx of slightly deleterious mutations because these are effectively invisible to selective pressures. It also asserts that beneficial mutations are too rare and too weak to counter it. So, these slightly deleterious mutations will accumulate in the population, having an increasingly deleterious effect on fitness after each generation, eventually leading to extinction. While creationists like plant geneticist John Sanford champion the concept today, it's nothing new in anti-evolution land. Genetic entropy was asserted as true by creationists even before there was any data that could actually be used to confirm or refute this position. I have a book from 1967 titled Did Man Get Here by Evolution or by Creation that claims organisms are breaking down over time, never gaining new functions or structures, and only acquiring harmful or neutral mutations. Therefore, this is, at its core, a doctrinal position that creationists have been scrambling around to provide data for half a century, rather than honestly evaluating the data as it comes in. As previously mentioned, genetic entropy relies on the premise that natural selection and the other mechanisms of evolution are too ineffective to stop the accumulation of deleterious mutations. These deleterious mutations don't have to be extremely outwardly deleterious, such as the mutation that causes PKU, but can instead merely be nearly neutral. A common analogy put forth is that this is similar to rust on a car. While the rust may be imperceptible at first, it will eventually overtake the car. So the question becomes whether this is actually happening in modern populations. It's important to understand that all mutations have some effect on the carrier, even if that effect is vanishingly small, as the 2007 paper, The Distribution of Fitness Effects of New Mutations, notes. The paper describes the fitness effect of mutations as either being 0, that is being lethal, 1 being neutral, and 2 being a 100% increase in fitness. So mutations that are very close to 1, like 0.999 or 1.001, are very close to neutral, therefore nearly neutral. And whether these mutations are nearly neutral or effectively neutral is a function of population size. In the founder effect, for instance, mutations have a much higher chance of being spread throughout the population than in a larger population. Now, let's get to the data. In his book on genetic entropy, Sanford relies on a complete mischaracterization of Moto Kimura's neutral theory of molecular evolution. In Figure 3c, he shows a mutation distribution graph that is adapted from Kimura's 1979 paper. This figure only includes a curve on the left for the deleterious mutations, but nothing on the right for beneficial mutations, since Kimura did not include them in his model. It should be noted that Sanford's adapted figure is slightly fudged. When you compare it with the original figure from Kimura's paper, you see that the no selection zone is significantly larger in Sanford's adapted figure. However, this is not the worst part of the misrepresentation. In Figure 3D of his book, with the title Correct Distribution, Sanford added a tiny curve on the right that represents the distribution of beneficial mutations, obviously keeping them extremely rare and very weak as they are all within the no selection zone. Sanford could have presented this as his own hypothesis for the mutation distribution, and there would be no problem with that, but that's not what he did. As Sanford said in his own book, quote, in Kimura's figure, he does not show any mutations to the right of 0, i.e. there are 0 beneficial mutations shown. He obviously considered beneficial mutations so rare as to be outside of consideration, close quote. And later, referring to the tiny curve that Sanford added himself, quote, what is most interesting about this figure is to realize that essentially the entire range of all hypothetical beneficial mutations falls within Kimura's effectively neutral zone. That means that essentially all beneficial mutations must be unselectable. So selection could never favor any such beneficial mutations, and they would essentially all drift out of the population. No wonder that Kimura preferred not to represent the distribution of the favorable mutations, close quote. So Sanford heavily implied that Kimura would agree with him on the distribution of beneficial mutations, even going so far as to say that Kimura would have drawn that tiny curve for beneficial mutations in the same way he did. But this is a complete distortion of Kimura's views and his model. In that theory, Kimura allows for the vast majority of mutations to be effectively or near neutral or harmful. However, beneficial mutations overwhelmingly overpower the former types. As Kimura himself stated, quote, note that in this formulation we disregard beneficial mutants and restrict our consideration only to deleterious and neutral mutations. Admittedly, this is an oversimplification, but as I shall show later, a model assuming that beneficial mutations also arise at a constant rate independent of environmental changes leads to unrealistic results, close quote. Further into the paper under the section Slightly Advantageous Mutations, Kimura explained what these unrealistic results were. When he tried to include beneficial mutations into his model, the rate of evolution could become enormously high, contrary to actual observations. In other words, Kimura omitted beneficial mutations not because he thought their impact was so small as to be outside of consideration. He did this because his model would collapse under the strong influence of beneficial mutations. The very opposite of what Sanford said. At the end of the paper, Kimura stated what his actual views are regarding the real-life impact of beneficial mutations. Quote, under the present model, effectively neutral but in fact very slightly deleterious mutants accumulate continuously in every species. Whether such a small rate of deterioration in fitness constitutes a threat to the survival and welfare of the species, not to the individual, is a moot point. But this will easily be taken care of by adaptive gene substitutions that must occur from time to time, say once every few hundred generations. Close quote. Sanford, being a geneticist himself, was therefore lying when he said, quote, population geneticists know that essentially all mutations are deleterious and that mutations having positive effects on fitness are so rare as to be excluded from such distribution diagrams, close quote. And anyone who had checked Sanford's claims could have found that out. With that in mind, the question becomes whether advantageous mutations do indeed exist, and the answer is overwhelmingly yes. Let's take some examples. For starters, garter snakes have repeatedly developed resistance to tetrodotoxins in their prey, the rough-skinned newt. Second, the water strider genus Ragavalia had a gene duplication that allowed the formation of novel propeller fans that gave it access to a new environment. Third, the Arctic cod Boreogatus actually turned a non-coding region of DNA into an antifreeze glycoprotein sequence utterly ignored by creationists. In each example, we have a genus or a set of species within a genus that have developed some novel structures through evolution. However, this isn't the only part of Sanford's argument. He also misrepresented James Crow's work when he claimed Crow said that all populations are slowly declining in fitness. Crow's work was only applied in that case to humans who are not under any strong selective pressures. It specifically applies to humans living in industrialized nations with low birth rates and high survival rates due to medical interventions. So it doesn't apply to all of humanity throughout our history, only to modern times and to those that are living under unique circumstances. This is the exact opposite of what Sanford is trying to argue, which is that natural selection is too slow to counteract the effect of deleterious mutations. If Sanford wants to support his own claims, he needs to provide instances of genetic deterioration in spite of selection, not in the absence of it. Another example of misrepresenting the work of scientists, Sanford added a graph in his book with the name Actual Biological Data Showing Mutation, Accumulation, and Fitness Decline in Human Influenza Virus to support his claims on genetic entropy. As Sanford describes himself, quote, "This graph illustrates the pathogenicity, i.e. fitness, of the H1N1 strain as well as the other two pandemic-causing strains during the last century," close quote. Sanford seems to think that the purpose of a virus is to harm or even kill its own host, causing devastating epidemics with high mortality rates. That's why he equates pathogenicity with fitness, but pathogenicity doesn't correlate with viral fitness. First, it's not always in the best interest of the virus to be highly virulent. The virus eventually needs to jump to a new host in order to survive. Thus, quickly killing or making its host too sick could prevent its own transmission. For a virus, higher fitness more often means being less lethal. The second reason for why pathogenicity doesn't correlate with virus fitness is that populations eventually acquire immunity, leading to a lower rate of illness over time, even if the virus copies its genome perfectly. In fact, populations will become immune especially if the virus doesn't change, but more on that later. In an attempt to support his claim of a pathogenicity/fitness decline in the H1N1 virus, Sanford references figure 1 from the paper by Simonson et al. For this specific figure, but the original paper doesn't say what Sanford claims it says. The original graph isn't about viral fitness nor even pathogenicity whatsoever. As Gerard Jellison points out in his review on Amazon, quote, "The point of the paper is that the flu epidemic of 1918 to 1919 was unusual in that it killed mostly young people. The authors studied how this ratio varied with time and modeled the results in terms of different immunity in the two age cohorts. Thus, the curve in Sanford's plot merely show that, as time goes on, flu strains kill a greater proportion of elderly people. This makes sense, since younger people have more vigorous immune systems. Sanford's figure is invalid, since it does not illustrate declining viral fitness at all. This blunder is an absurd misuse and/or misunderstanding of scientific data." Close quote. Sanford also frequently claims that the original 1918 strain became extinct as the result of genetic entropy, at least in the human lineage. As was just pointed out, the data that Sanford references does not actually support his conclusions. But there is more. The virus isn't technically extinct. While the original strain is gone, the 1918 virus left descendants that led to new strains through processes such as antigenic drift and shift, which were mentioned in the previous video on evolutionary medicine. Viruses are constantly evolving into new strains in order to avoid our immune system, and the descendants of the 1918 virus are still causing misery to this day. As the paper with the title 1918 Influenza, the Mother of All Pandemics notes, quote, "All influenza A pandemics since that time, and indeed almost all cases of influenza A worldwide, excepting human infections from avian viruses such as H5N1 and H7N7, have been caused by descendants of the 1918 virus, including drifted H1N1 viruses and reassorted H2N2 and H3N2 viruses. The latter are composed of key genes from the 1918 virus, updated by subsequently incorporated avian influenza genes that code for novel surface proteins, making the 1918 virus indeed the mother of all pandemics." Close quote. It should be mentioned that most laboratory experiments do not support genetic entropy, a fact that John Sanford has acknowledged on record, although he simply provides excuses for this inconvenient observation despite making his position even less tenable. It should also be noted that there are experiments, such as the long-term E. coli experiment, which have borne out data that is unfavorable to genetic entropy. The 2015 paper, Sustained Fitness Gains and Variability in Fitness Trajectories in the Long-Term Evolution Experiment with Escherichia coli, indicated that there is no upper limit to organismal fitness and that the bacteria have been increasing in fitness throughout the course of the experiment in 12 separate populations, no less. Creationists might whine that this experiment took place in the lab. However, the fact is that this shouldn't be happening at all under the genetic entropy model. Sanford specifically has said this about the Lenski experiment.
For example, all of the beneficial mutations in the long-term Lenski experiment were loss of function. Genes were deleted, genes were silenced, or genes were downregulated. So in one case, a promoter, which is normally regulated, became an unregulated promoter. But all that is adaptive fine-tuning. There was no new information being created. And in fact, technically, as the bacteria jettisons unnecessary genes for that environment, they are actually painting themselves into a corner. They won't be able to survive in any other environment.
Setting aside the fact that not all beneficial regulatory changes were the result of genes being deleted, silenced, or downregulated, some genes were upregulated. Also, the most famous beneficial mutation that led to the CYT plus phenotype was the result of a tandem duplication, which formed a novel regulatory module by placing a copy of the CYT gene under the control of a copy of the RNK promoter that is activated under aerobic conditions. Not exactly a loss of function. And setting aside the unsupported claim that these bacteria are no longer able or less able to adapt to a different environment, Sanford blatantly dismissed the observation of a continuous increase in fitness among E. coli by asserting that their genome was still deteriorating, but with a catch. Only the expendable part of the genome was deteriorating, which apparently allows for an indefinite increase in fitness, which Sanford calls adaptive fine-tuning. However, he gives no explanation for why the vital part of the genome didn't deteriorate during the Lenski experiment. What does genetic entropy even mean at this point? Does it only deteriorate the expendable genes at first, allowing for an unquantified short-term increase in fitness? And once that is all gone, only then will it affect the vital genes and cause a decline in fitness? If so, when is that turning point? For how many generations do we need to keep an eye on E. coli until we can finally observe a decline in fitness? Sanford hasn't explained exactly how genetic entropy is supposed to operate in this manner, leaving it rather ambiguous to what we should or shouldn't expect from it. He even attempts to shift the focus onto new information, a creationist trope that is even more ambiguous. All of this makes it clear that, when faced with evidence to the contrary, Sanford is simply moving the goalpost. And Sanford's model must apply to paleogenomics, but it doesn't in any way support his case. For example, paleogenomic and paleoproteomic studies have been done on mammoths, mastodons, glyptodonts, the American cheetah, saber-toothed cats, giant sloths, toxodon, macrochinia, stilt-legged horses, Neanderthals, Denisovans, archaic humans, and other organisms, and yet there is no sign of increasing genomic degradation as time goes on. On a final note, I would like to draw attention to this particular paper called Dynamic Mutation Selection Balance as an Evolutionary Attractor. It notes that since asexual populations are subject to Muller's ratchet, they are accumulating deleterious mutations which cannot be completely prevented by purifying selection. The ratchet refers to the fact that this process is irreversible. When a deleterious mutation is fixed in the population by genetic drift, the most fit genotype is lost. Consequently, Muller's ratchet leads to a rapid decline of population fitness, especially if the deleterious mutations are weakly selected. Therefore, maintaining the fitness of asexual populations must require an influx of beneficial mutations, which continuously compensate for the accumulation of deleterious ones. And since beneficial mutation rates increase as fitness declines, eventually the influx of beneficial mutations becomes high enough to counter Muller's ratchet. Once that happens, the population remains in a stable dynamic equilibrium, as illustrated in the first figure of this paper. It also neatly shows that poorly adapted populations will experience an excess of beneficial mutations that will push it towards higher fitness. Conversely, in an over-adapted population, deleterious mutations dominate, reducing the population fitness until the balancing point is reached. Furthermore, since selection favors beneficial mutations, this balancing point is put at a high level of fitness, making it possible to maintain a well-adapted population. This mutation selection balance point is described as an evolutionary attractor, something that shouldn't exist if there is any truth to the concept of genetic entropy, but spoilers. As the paper reads, quote, "We argue that such a state can exist for any population size n and mutation rate u and calculate the fraction of beneficial mutations epsilon that maintains the balanced state. We find that a surprisingly low epsilon suffices to achieve stability even in small populations in the face of high mutation rates and weak selection, maintaining a well-adapted population in spite of Muller's ratchet. This may explain the maintenance of mitochondria and other asexual genomes," close quote. And it also says, following in reference to laboratory experiments, quote, "Examining the properties of the dynamic mutation selection equilibrium over the full parameter range, shown in Figure 6, has revealed a strong asymmetry between beneficial and deleterious mutations. For example, for a small population, n times s is approximately 10 to the third, with high mutation rate lambda is approximately 8. Just 2% beneficial mutations are enough to counteract the effects of deleterious mutations. This indicates that purifying selection is remarkably effective even for conditions where Muller's ratchet would proceed extremely quickly in the absence of back and compensatory mutations. Populations with epsilons greater than epsilon times c should adapt, while populations with epsilon is less than epsilon times c should decline in fitness. Experimental evolution of model organisms in controlled laboratory environments appears to be consistent with this expectation. In the long-term evolution experiments of Lenski and collaborators in Escherichia coli, parameters are such that we expect the critical epsilon c to be very small. Epsilon c is much less than 10 to the negative 10th, consistent with their observation of continuous adaptation even after tens of thousands of generations." Close quote. In summation, Sanford has utterly failed to provide evidence for his genetic entropy model, as it isn't supported in modern humans, paleogenomic samples, long-term bacteria experiments, or even the H1N1 model that he attempted to apply it to. So, thanks for watching, and I'll see you next time.